Okay, good morning, everyone. Today we have one presentation by Mathieu Lavallee-Adam from TSRI. He's going to talk about PSEA-Quant and go over some updates since the last presentation. If you missed his last presentation, and you want to, if you want to catch up on it afterwards, the YouTube link has been emailed out. So just a reminder, this session is being recorded, and this will also be uploaded available on YouTube shortly after the conference. So I'll turn it over to Mathieu.
So thank you very much for the introduction. Good morning or good afternoon, everyone. So indeed, my name is Mathieu Lavallee-Adam, and I'm going to present some of the updates of PSEA-Quant since our last technical meeting where PSEA-Quant was presented. And I should mention that some of the work for these updates has been done by other members of the Yates Lab, Salvador, Martinez-Bartolome, and Robin Park have been in close collaboration with me to do some of the work that will be presented here. So this is work, as I said, done in the Yates Lab at the Scripps Research Institute. So since some of you might have missed or might not remember everything regarding PSEA-Quant, I thought I'd just do a quick background review of the idea behind PSEA-Quant. And so basically, one of the typical ways that people end up analyzing their data sets in order to do some sort of functional enrichment analysis is the typical or traditional gene ontology analysis strategy. And there are a variety of tools that do this, from David, Gorilla, Ontologizer, GoStat. Some of you might be using the Ingenuity framework in order to do some sort of gene ontology or functional enrichment analysis. And the goal of these analysis is to perform a statistical assessment of the overrepresentation of Go terms, which can be basically molecular functions or biological processes or cellular components in a given data set. And this is often done in the context, for instance, of proteomics or even mass spectrometry-based proteomics. This is often done by selecting somehow a set of significant proteins. So, for instance, proteins that are meeting certain abundance criteria or certain confidence criteria, and evaluate if among these significant proteins there is an enrichment for a certain molecular function or Go process. So basically, if the proteins in red here that are annotated with a Go term are actually overrepresented in the set of significant proteins based on their overall representation in the entire data set. But these analyses have certain limitations. One of the main ones is probably that it assumes that the gene or the protein expression is independent. And that's something that we know is not exactly true, since, for instance, if we have proteins that are members of the same protein complex, it happens quite often that these proteins are regulated in the same fashion so that they are co-expressed in order to co-occur at the same time in the same cell and the same compartment of the cell, I'm sorry. And this is an example that can be given from yeast, for instance, where we have subunits of the ribosome that are all regulated or using the same transcription factor. And so this is an assumption which often fails when you're doing a gene ontology analysis and causes some issues. Another limitation is that it does not consider gene or protein expression abundances. So basically, once you're in that set of significant proteins, if you're a protein that has a Go term that you are analyzing, if the protein is of high abundance or of low abundance, it doesn't really matter. It's a binary count of the overrepresentation of that protein. It's whether it's close to the threshold or further away, it's treated in the same way. It requires the establishment of arbitrarily chosen thresholds. So, for instance, I basically drew a line somewhere at the abundance that I considered that was enough to be significant, and I drew a line at the confidence that I considered was enough to be significant. But these thresholds are fairly arbitrary, even though there are some guidelines in literature in order to choose them. the problem is that if two proteins are very close to that threshold, one above, one below that threshold, they end up treated very differently, even though their values are very close. Again, that idea of you having a binary count for overrepresentation becomes a limitation here. And two final limitation is that because of all these assumptions that are often violated, the gene ontology analysis often yields long lists of enriched goal terms that are difficult for the user to parse and interpret. And also the methods are not that well adapted for quantitative proteomics data and some of its features, such as the reproducibility of the data. So for all these reasons, in the Yates lab, we were interested in trying to provide a tool that would be more interesting for quantitative proteomics analysis, and we therefore developed PSEA-Quant. And the goal of PSEA-Quant is to basically statistically assess the overrepresentation of protein sets. So again, biological processes, molecular functions, or disease associations. And so we want to assess the overrepresentation of these protein sets among proteins that are of high abundance, and for which the abundance measurements have a good reproducibility, so i.e. low coefficient of variation, in a data set that is composed of a single or multiple conditions measured through mass spectrometry. And so this is a GSCA-inspired method, so gene set enrichment analysis-inspired approach, which is adapted for proteomics. And basically, and I should mention that this is published in the Journal of Proteome Research about a year ago now. And the goal of PSEA-Quant, or the way PSEA-Quant works, is if we look at this toy example of a data set, we basically have a quantitative proteomics data set where we have 3 replicates, and we have along the heat map, we have 7 proteins that represent our toy example or a fictitious protein set. And PSEA-Quant basically scores that protein set by looking at the average abundance of these proteins in the heat map that we have here among these replicates, and look at the coefficient of variation of these abundance measurement for each of these proteins. And based on these values, it maps each of these proteins of the protein set into an enrichment score weight matrix. So proteins that are of high abundance, for instance, P1 in that example, and that have a low coefficient of variation, such as P1, obtain a very good score because they're the proteins that we're interested in. And we can see that each of these proteins obtain a different score in the abundance weight matrix, and we therefore, in order to score the protein set, sum up all of the individual protein scores and obtain a final enrichment score for a given protein set. So protein sets that are enriched with proteins of high abundance and low coefficient of variation will obtain high score. We then have a Monte Carlo sampling procedure that assesses the statistical significance of the scores that are obtained, the enrichment scores that are obtained for each protein set. So I'm not going to go into the details of that analysis because that was presented in last time. And as was said in the introduction, can be viewed on the internet or referred to in the JPR, the Journal of Proteomics, Proteome Research paper. So what I'm going to focus on today is some of the advantages of the approach and how we've did some of the updates to highlight some of these advantages. So the advantages of PSEA-Quant is that it can be applied on samples that are originating from a single or more conditions, and we facilitated that input over the last few months. And does not require any of the arbitrary threshold because it basically looks at the enrichment overall in the data set of these proteins. So that's actually pretty useful, where the user simply has to input the entire data set and does not need to choose thresholds for the analysis. And it models the likelihood of a protein to be annotated by a certain protein set and protein abundance correlations. I didn't go into the details, but this is all in the Monte Carlo sampling procedure that is described in the YouTube presentation and the paper. So some of the new developments of PSEA-Quant, I have a brief list here of the development, and I'll go into the details individually for all of them. But so to list them, we developed an API in order to link PSEA-Quant with some of the other tools of the center. We've also added new organism for the enrichment analysis. We've integrated new additional disease annotation databases, so that provides more protein sets, so that we have more than just molecular functions and biological processes, for instance. We have additional input features, which I've hinted before, so to cater to more PSEA-Quant users. And we've also integrated PSEA-Quant in some of the external tools that was developed in the Yates lab. And there's a few miscellaneous additions that I'm going to talk about at the end. So the development of an API, so basically Salvador Martinez-Bartolome has developed an easy-to-use API. It's actually very simple, that allows us to basically do HTTP requests to the PSEA-Quant server. We are currently polishing that API, but it is currently available upon request, but we will distribute it and put it on the PSEA-Quant website as soon as possible. And the goal here is to allow any tools, obviously, to be able to link the data that they contain to, or the data that was input in the tool, so that they can actually perform a PSEA-Quant request in a more in an easy fashion. And that was something that was discussed a few times when we met with the other members of the center in order to facilitate bridging the gap between the tools that are developed in the BT2K initiative. Some of the new organisms that were added, we were already allowing the analysis of human, rat, and mouse with PSEA-Quant, and then we have now added Drosophila melanogaster, or fly, and yeast, Saccharomyces cerevisiae, based on user feedback. So we constantly listen. We have a good pool of users, and we constantly listen to their needs, and they do not hesitate to ask if the analysis can be performed on a certain organism. And so we've been adding these in order to cater to their needs. And we're currently in the process of including E. coli and zebrafish as other organisms that can be analyzed by our tool. And we think that with this, we will have a good pool of classic model organisms. But we'll see if there is a need for more organisms. Maybe another that we were considering is also Rhodopsis as a plant model organism. We'll see if there's more that comes up or that are requested. If you have any suggestions, I will also be listening to these. Regarding disease association databases, I am currently working with Howard Choi from the PINGS lab, and so working currently integrating the COPA-KB database, so basically cardiovascular diseases associations, into the PSEA-Quant analysis. And one of our future plans is also to include the online Mendelian inheritance in men, or more commonly known as OMIM database, in order to augment the number of proteins or disease-related protein sets that can be analyzed with PSEA-Quant. New input features. So we've actually facilitated the analysis of relative abundance quantification data set. And what do I mean by that is that PSEA-Quant allows the analysis of data sets that are analyzed, that are quantified using label-free strategies, or mass spectrometry label-free strategies, or more formally known as spectral counting or intensity-based measurement of protein abundances. But it also allows the relative quantification analysis, where we would have two data sets of different conditions that are compared, and so where we basically, for each protein, have some sort of abundance ratio. So this can be done either through a label-free strategy or using some isotopic labeling. And in these strategies, the user will have, for each of the proteins in the data set, an abundance ratio between two conditions. And the proteins that would have high ratios, basically proteins that are more expressed in condition A versus condition B. So you before that actual input improvement, if I can say, of PSEA-Quant, the user that would have a relative abundance quantification data set, if he wanted to do a PSE-Quant analysis, would basically have to submit one data set including the ratios of condition A versus condition B to see the proteins that are enriched in condition A. And then he would have to inverse the ratios and resubmit the analysis in order to see the protein set that would be enriched in condition B versus condition A. So we facilitated this process by automating it and allowing the user to basically select an option so that everything is done concurrently, and he doesn't have to do that extra manipulation. So we think, again, that we're catering to the needs of our users by doing so and making PSE-Quant a little bit more easy to use for them. We've also added new types of inputs in terms of absolute quantification. So I was talking about spectral counts before, but now we can also analyze data sets that are quantified using normalized spectral count strategies such as NSAF. And we've also added PSE-Quant module for the analysis of proteins that would be quantified using ion intensity values. These are basically this is another proxy that comes from the mass spectrometry analysis for the protein abundance. We've also integrated PSE-Quant to external tools that are developed in the Yates lab and by integrated proteomics applications. So PINT, which was presented in a technical meeting, BD2K technical meeting before, PINT stands for Proteomics Integrator. And Proteomics Integrator is basically online experiment repository where a given lab can store all of its mass spectrometry-based results in order to integrate them, but also query them using some sophisticated queries, so to see if a protein came up in different experiments from different users. So everything is integrated into that repository, and we wanted to be able to analyze the data sets in that repository with PSE-Quant without having to download them and resubmit them to PSE-Quant. So Salvador Martinez-Bartolome created an API and linked PINT with PSE-Quant so that the data set that are stored in PINT can be easily analyzed with PSE-Quant. Similarly, IP2, the integrated proteomics pipeline from integrated proteomics applications, is on the verge of launching a PSE-Quant module. It's virtually completed right now. That allows all the data sets that have been the mass spectrometry-based proteomics data set that have been analyzed in IP2 to also be analyzed with PSE-Quant. And both of these tools are basically replicating the web interface of PSE-Quant, so users that are familiar with one of the interface can actually be doing the transition very easily. And so the IP2 integration of PSE-Quant was done by Robin Park, and I thank him also for that. As a future integration, we are currently working with Howard Choi to integrate PSE-Quant in the COPA-KB, the Cardiac Organeller Protein Atlas Knowledge Base, in order for the data sets that are stored in COPA-KB or inputted into COPA-KB to be also analyzed with PSE-Quant if the user wants to. And so this is work in progress, and we hope to be able to do so very soon. Yeah, and I should mention that the creation of the API was crucial to obviously to the integration in all these tools. So I'd like maybe another miscellaneous additions. Now the output emails that PSE-Quant gives, because when the PSE-Quant run is completed, an email is sent, since the run requires a fairly lengthy Monte Carlo sampling analysis. Well, that output email now provides more details about the analysis. That was something that our user was requesting, such as the parameters, some of the extra parameters that are selected. And we also provide more tips on how to analyze the data in that email. And we've also submitted the protocol manuscript in Current Protocols in Bioinformatics on September 15 of this year. We think that this will actually be very useful for some of the more novice bioinformatics users to understand what option to select with PSE-Quant and how to interpret the result and how to troubleshoot a certain data set that was analyzed with PSEA-Quant. We think that this can actually help users getting around the tool and providing details or more technical details that were not provided in the original publication of PSEA-Quant. And we hope that this will obviously be published fairly shortly. Some of the future work regarding PSEA-Quant, we are looking at obviously, as I said, as the addition of the COPA-KB and OMIM disease association database for the analysis of PSEA-Quant. Another important aspect is that we're looking at the addition of an algorithm for protein set similarity-based clustering. And what do I mean by that? Is that a lot of the protein sets that can be reported by PSEA-Quant might be redundant if they include several of the same genes. They will basically, if one of the protein set is significant and another one that includes similar proteins will also very likely to be significant. And in order to help the user parsing and making sense of the output set list of significant protein set of PSEA-Quant, we would like to be able to cluster them together without too much of a loss of information. And so we're looking at different strategies that have been proposed, and their goal is to either integrate one of them or design something inspired by the work that was already done in order to provide a more intelligible output for PSEA-Quant. We are also, as I said, looking at the addition of more organisms for PSEA-Quant analysis, so E. coli and zebrafish. And basically, our future work is guided by user feedback. So we're continuously going to improve based on user feedback from what PSEA-Quant has been used. I should mention that PSEA-Quant has been used more than 500 times despite its recent publication, and fairly small user base, I guess I could say, of the quantitative proteomics world. And so we're quite happy about our users' usage right now and the feedback that we get from them. Before I end, I'd like to thank my supervisor, John Yates, Salvador, Martinez-Bartolome, and Robin Park, with whom I've worked in close collaboration the last few months to improve PSEA-Quant, all of our PSEA-Quant users, and the funding from the NIH and the Quebec Research Fund. And before I completely end, I guess I'd just like to show you a few additions to the PSEA-Quant interface. The PSEA-Quant interface is fairly rudimentary, but it doesn't need to be much more complicated than that, I guess. So some of the new things since last time, we basically have an option to select the quantification type of the mass spectrometry-based proteomics analysis, and the abundance ratio with the addition of the inverse ratio is now an option for the user. The organisms now add an addition of mouse, rats, and human, the Drosophila melanogaster, and the yeast organisms that can be used for the analysis. The input data set are inputted in the same fashion as before. There's no change to the Monte Carlo sampling algorithm, or basically the statistical assessment algorithm. The core of the software is not changed, so all of these parameters did not change, but we also have the addition of the cardiac organular protein atlas knowledge base, or COPA-KB, as the annotation database that can be used with PSEA-Quant. And the user can, at the end, select some of the Monte Carlo sampling algorithm parameters and input his email to receive the email when the PSEA-Quant analysis has terminated to get his output. So the interface is fairly simple. It might be in the future plan to make it a little bit more exotic, but for now we find it convenient and basically provides all the information the user needs to have in order to do a PSEA-Quant run. And finally, as I said before, PSEA-Quant is available at that web address, but it's also available with Pint and IP2 tools that are developed in the Yates Lab. I thank you very much, and I'll be happy to answer your questions.
I would just like to offer the recommendation for the API when it's released, the Swagger UI. you're probably familiar with that from mygene.info. But yeah, we've been using that for CopaKB's API, and we think it's a really good way to release release documentation it's really easy to use and really easy for users to build queries.
Okay. Yeah. Perfect. Yeah, that's a good idea. We'll definitely look into that.
So this is Andrew here. Yeah, this is Andrew. Can you hear me?
Yes. Yes.
Mathieu?
Hi, Andrew.
Great. Hey, so nice presentation, of course. So question on adding new organisms. What is it that is the work required there? What's the rate limiting step?
The limiting step is the synonyms, basically, of all the protein symbols. The annotations, for instance, so that one of our main usage of PSEA-Quant is still Go annotations, and Go annotations do not necessarily always use the same identifiers for all of the proteins, and I guess you're pretty familiar with that. And so we're trying to make it as easy as possible so that the user can input as many type of identifier as possible in order to get as many annotations as possible from the Go database. And so it's always getting the synonyms tables to work and not being redundant, but also the more comprehensive as possible. So it's not that much work. It's just that we like to do things properly regarding that. Yeah, that's pretty much it, I guess.
Makes perfect sense. The only thing I think we could explore whether or not mygene.info could be a resource there.
Yes.
You know, the synonym resolution and identifier resolution and gene ontology retrieval and all this, I mean, is one of the core use cases of mygene.info. Obviously, you need to, you know, just for performance reasons, you can't do everything through the mygene.info API. But, for example, translating user queries, various identifiers, into some canonical identifier that is related to things in your database, that is something that I think mygene.info should fit in that niche.
That's true, and actually I didn't think about that, and that's a very good point. And we could basically use the API when the input data set is used and then deal with it. Yeah, that's a very good point, Andrew. Thank you. We're going to look into that.
Great. I had another question on the gene disease annotations. Maybe this is just a suggestion of another resource to look into. You mentioned you were going to add in CopaKB and OMIM. There is a group that we work with at University of British Columbia developing a resource called PhenoCarta. And their goal is to aggregate gene disease associations from a variety of different resources, including OMIM and specialty ones like ALS genes for Alzheimer genes. There's a variety of databases here I'm looking at that the vast majority of them I've never heard of, but GWAS catalog is another one. They don't have CopaKB in there, but so I guess my suggestion might be twofold. A, let's figure out how to get CopaKB disease associations in there, and then B, you could use them as, again, a resource to aggregate gene disease associations from a variety of resources, so you don't have to continually add them piecemeal.
Right. No, that I wasn't aware of this. You called it. Can you repeat the name? PhenoCarta. Is that how you called it?
Correct. Pheno as in phenotype, and Carta, I don't know, Magna Carta.
As in Carta.
Yeah.
Okay, excellent. Wasn't aware of that, and yeah, that would be obviously a lot easier than to add the list of 20 these disease gene and association database. So yeah, no, you know which group at UBC who's taking care of it?
It's Paul Pavlidis.
Okay.
If you have any problems finding it, just get in touch. We work with them pretty closely.
Perfect. That's great. Thanks again for the input, Andrew.
Yep. And if nobody has another thought, I'm going to have one more thought. So the integration with Pint sounds really cool. I would like to see that at some point. But it makes me wonder whether a similar integration could be envisioned down the road with PRIDE.
I don't see why not. Yeah, I don't see why not. I mean, Pint is not a repository. I mean, I think you are a little bit familiar with Pint. It's not a repository as in PRIDE. The goal is not to basically store all of the data set in the world, but it's more like your own data sets from your lab. But there's no reason why we couldn't integrate it in PRIDE. Yeah, no, I'd be definitely open to work.
And you can imagine that either, and realizing on the EBI side, they need to be very, a lot of thought goes into what features are there, but through this you might, I don't know, this is a malformed thought, but you might think about how to, have an easy import. So it's like, paste your PRIDE ID here, and then we'll come and import it and get the gene lists or protein lists and things like that.
Yeah, no, yeah, definitely. Yeah, I would assume like we would have a few things to look in that in terms of the identifiers again, and making sure that these things are following the right formats or the right identifiers. But I think I would be very much happy about that for sure.
This is Henning. I was actually about to say the same thing, and one of the things we should look into is to use the interface we are planning to develop within the context of BD2K anyway, which is meant to retrieve data sets, proteomics or even metabolomics, or so expression data sets from resources for exactly this kind of further processing. This is the proteome exchange, or in a wider sense, the XI interface, which we hope to, or which we have started working on, but that is by far not yet finished, but that would hopefully provide a standardized interface to do these things. And it's exactly meant for this kind of communication.
Right. No, for sure. So we should keep in touch regarding this, and yeah, I'd be very much interested in working on this.
Another just question you mentioned, it takes a while, and that's why you're sending an email notification. So what is the typical analysis duration for, let's say, a typical proteomics data set of 10,000 computations, which is fairly high resolution, let's say 5,000?
On average, we're looking at 1 to 2 hours. It can be much shorter. We can be in the order of 15 minutes, and it can be much longer. We can be in the order of almost a day. It depends a lot on the sampling options, the Monte Carlo sampling options that are selected by the user. So, for instance, some users will want very high accuracy for the significance p-values that are outputted, maybe because their data set is a little bit smaller, and in order to do so, the more sampling is required, which is equal to a larger running time. So the running time is dependent on that. It's dependent also on the type of annotations that the user is looking at. The molecular signature database is significantly bigger than the gene ontology database. There's a few of these things, but we're talking about 1 hour on average. That would be the short answer.
Thank you.